Hello and welcome to today's webinar, Selecting the Right Clinical Trial Design, Impact and Probability of Success, and Power. Today's webinar will provide you an overview of some of the common choices and decisions available when designing your clinical trial, some guidance on what the best decisions might be for your trial, some common pitfalls that occur in many real trials, and then talk about how statistical power can be one metric that you can use to both decide between different designs and also communicate why one design choice may be superior to another. Before we go any further, just some frequently asked questions. Is this webinar being recorded? Yes, the recording and the slides will be sent to you after this webinar is complete via email. If you have any other questions, please add them into the questions tab available in your webinar software. We may not have time to answer them live, but these all will be answered via email after the webinar is complete. So without any further ado, welcome to today's webinar, Selecting the Right Clinical Trial Design, presented by nQuery. As I mentioned, the hope is to provide you a lot of really useful guidance for talking about and thinking about how to design the optimal trial for your particular scenario that's of interest, and then talk about statistical power as one way that you can help make those decisions and also talk about those decisions with other stakeholders. My name is Ronan Fitzpatrick. I'm the head of statistics here at nQuery. I've been over the company over a decade, and I think clinical trial design is one of those areas where, despite the fact that there's a lot of guidance out there and there's a lot of metrics that you can use to make the right choice, there's still a lot of cases where bad choices or suboptimal choices are made when designing clinical trials. So in terms of what we're going to cover today, first we're going to talk about clinical trial design at a high level, kind of go through many of the top choices, many of the top considerations you should have when thinking about designing your clinical trial, and then also talk about some of the common pitfalls that occur in many real trials. We'll then talk about statistical power as one metric you could use to decide or communicate why you should make certain choices in a clinical trial, and then have some practical demonstrations of using power calculations for that purpose. At the end, we'll have time for conclusions and discussion. So today's webinar is brought to you by nQuery, the complete solution for optimizing your clinical trial designs, which you can see covers kind of sample size, adaptive design type calculations from preclinical research up to confirmatory phase 3 trials, and indeed actually into phase 4 trials, post-marketing surveillance as well. We've actually made a major update at nQuery just last week, which includes a table select or feature select overhaul, which should hopefully make it a lot easier and a lot more coherent for finding the right option for your particular scenario. And I'm going to show that in depth during the demo later on, but that's something to look forward to there. Just see where nQuery is a tiered product, where if you have the top tier expert, you get all the prior tiers as well. And base covers our sample size and power calculations for fixed term trials. Plus covers our Bayesian sample size features. Pro is for our adaptive design features, and expert covers our milestone prediction simulation features. There's a URL in the slides that will be sent to you afterwards, where you can check out what's in each of these tiers in detail. So let's get right into part one, the clinical trial design primer. So as we know, clinical trial design is a huge area, and it's obviously been of interest since the first time clinical trials started to become the norm. Obviously, we go back all the way back to the late 19th century, early 20th century, people like Ronan Fitzpatrick and Gossett, and talking about trial design and the concepts behind that, and that kind of emerging, especially in areas like agriculture, up onto the mid 20th century. And then we saw this explosion in clinical trial use in medicines in the mid 20th century that's only continued to evolve since then. But I think even in the last 10, 20 years, we've seen a much even rapider increase in the evolution, in the openness to innovation in the clinical trial space, especially at confirmatory trials. I think phase 1 and phase 2 have had that evolution happen even faster in the background, but in recent years, especially since the COVID-19 pandemic, I think clinical trial design, even in phase 3, has started to evolve and adapt much faster than we would have previously been used to. And so when we're talking about complexity, we're talking about adaptive design, Bayesian analysis, integrating real world data, all of these considerations. Now, you may be asking, or look at kind of context of complex designs and stuff like that, why are we then going to today talk about the basics or the principles of clinical trial design? And what I would argue is that the principles are even more important than ever, because as you get more complex, it's easier and easier to get abstracted away or to forget about what are the basic principles on which you're building your clinical trial. Because obviously the clinical trial is designed to answer scientific questions. Now that might be in a commercial context, where the scientific question is, does your therapeutic work sufficiently that it should be available to actual patients in the real world? But that could also be like an intervention or comparing treatments that are already approved if you're talking about an investigator-led trial, for example. And poor design choices so often can lead to rejection or to generating poor evidence, and the negative conclusions of that can be seen all the time in the real world, whether that be drugs getting approved that shouldn't have been approved, or whether that be interventions or confusion about what the optimal decision could be to improve the lives of patients in the real world. And so what I believe is that thinking through design choices can provide value about not just the science and the statistics, but also improving the experience of patients within a clinical trial. And they're one of the stakeholders that I think until recently were often ignored, but I think patients are one of those ones that have the most powerful insights into what would actually get people to be interested in going to a trial, staying in a trial. And so, thinking about our clinical trial design from the ground up with them in mind also makes a lot of sense. And so we're going to focus here on the basic principles that should inform clinical trial design, and then talk about some common pitfalls and things that you can avoid in your trial, and then talk about power as one way that you can communicate that to other stakeholders. Because if you have low power, that either implies that your trial doesn't really have enough probability of finding for the conclusion that you want, even if that conclusion actually is true, or alternatively, if you're doing a sample size calculation, the massive increase you might see in sample size by making that pitfall in your trial. So let's go through the considerations here in a checklist, and like this kind of replicates checklists that you might see from other areas. So I know the Consort and Spirit guidelines are really, really, really useful guidelines. They're in the URL for those is provided the references at the end of the slide deck, and those basically cover both the protocol and the reporting of a clinical trial. So I would say that those are probably even more detailed than we'll go through today, and very much going through if you want to have a checklist of what you should do in your clinical trial, and that really helps then you think through what the clinical trial is. But we'll kind of take a more, maybe a slightly looser approach to this and kind of do to kind of go from the bottom up in terms of what we should be considering. So the first thing, and it's a very obvious thing, and it's kind of so broad it could almost be a webinar by itself, but the first thing you should be thinking of is what is the objective of your trial? So what are the scientific questions or questions that are of interest? Now, in a commercial clinical trial, this is usually pretty obvious. Your question is our therapeutic, whether that be a drug or a vaccine or a biosimilar, are they effective? And that efficacy could mean different things, like it's similar to an existing drug with a biosimilar, or that it's more effective or than an existing drug if it's a new therapeutic, or for a vaccine that this provides a protection against a disease of interest if applied to the population. And then if you're going beyond, say, just like commercial clinical trials to investigator-led trials, you may have, like, do we want to explain something or do provide inference, or are we also interested in having a prediction? And so prediction models itself is a huge, wide area with its own sample size requirements, and that basically should be built into what you're thinking of when you think about the rest of these questions as well. And you, as I said, you should be thinking about, like, what do I want my intervention to do? Do I need it to be better, worse, no worse than whatever the existing or standard treatment is, or we even have to think about the control group. Should that be, if we have a control group, is that a placebo or is that like standard treatment? And like, what is the objective we're trying to prove? Like, if there's already a well-established efficacious treatment for a particular disease, if we're going to get a new drug approved that doesn't have any major advantages in terms of being easier to administer or cost or something like that, then we're probably going to need to establish if our drug is superior to that drug, which we call inequality or superiority hypothesis. And then in certain cases, like a vaccine trial, you may want to establish not just that your vaccine is equivalent, but you may need to show that it's better than by a certain pre-specified margin. And so I think that really kind of feeds into, like, here's the question I will answer, and then obviously, like, who am I going to answer them for? Now, if you're doing an investigator-led trial, you may be interested in answering the question for yourself in terms of being a scientist or working with a scientist. But in many cases, there are obviously other stakeholders who you should be keeping in mind. So obviously, if you're trying to get a new therapeutic approved via the regulatory process, you're obviously going to have to keep the regulator in mind, and you're going to have to keep in mind what phase of the trial that this trial is for. So there's very different requirements for phase 1, where you're just trying to establish safety, at phase 2, where you're just trying to show proof of concept, and phase 3, which is confirmatory. And the regulator will have a much greater interest in, say, the type 1 error at phase 3 than they would at phase 1 and phase 2, where most of the risk is really to the sponsor rather than the regulator. That doesn't mean there's no risk to the regulator. They certainly will ask for some control, but phase 3 is where the regulator is going, okay, if you can get past phase 3, that's basically when the new drug application actually gets to a point where actual patients could be getting it outside of the trial context. And if you're talking about trying to communicate to patients or to scientists or other stakeholders, you should try and create a trial which is most likely to be persuasive to those other stakeholders as well. And so think about the priority audience for your trial, and that should help then change how you may run the trial. And obviously, they're maybe not the audience per se, but the patients who are going to be in your trial are part of the trial itself. So they should always be at least part of your audience consideration, in the sense that you need those people to want to join your trial, to understand your trial, and then also to want to continue to participate in your trial, because we know that non-adherence, people dropping out, is such a huge problem in many, many clinical trials. And then there's obviously practical constraints. If you're talking about a rare disease, then obviously patient recruitment will be much more challenging than for a common condition like, say, diabetes. And then obviously your time and financial considerations are going to be much more important if you're running like a public interest trial or like an investigator-led trial, than for a commercial trial, where obviously at the point you get to phase 3, a huge amount of investment's already up there. So the cost of the phase 3 itself is obviously not a no consideration, but maybe less of consideration. And then obviously you need to take into mind how much expertise do you have available to you, and kind of take that into mind in terms of doing what you're going to do the trial. Because if you only have like yourself, or you're just running it, you and one statistician, or just by yourself, like that kind of scenario, you may want to simplify down and use a more tractable trial than getting into like a complex multi-arm Bayesian type trial with hierarchical features and such. And so I think once you've thought about what the objective of your trial is, the next thing you think about is like, okay, I'm going to gather data. How am I going to gather this data, and what kind of data will be collected? And so, the main thing is you're going to go, okay, it's very tempting when you have a clinical trial, just say, I'm just going to measure everything. But that often makes, that often includes a cost, because obviously, if you're asking loads of questions, you're getting a lot of metrics, or you're taking blood measurements and stuff like that, all implies an extra cost, an extra time, and an extra burden on patients. So unless you can scientifically justify that, that is something which you should be avoiding, unless you, as I said, you can find it's either trivially easy to find, like say gender, or it's justified by this. We think this would be a useful biomarker, or we think this would be a useful secondary endpoint to keep in considerations. And so you need to be able to justify all of the data you're going to measure if you're really going to well consider your trial. And obviously there are legal and ethical constraints depending on the jurisdiction that you need to take into consideration as well. And obviously something that you should be talking to the relevant like legal authority or regulator, or to a lawyer yourself to make sure you're on the right thing. And then once you've got past that and you're like, okay, I'm going to collect this type of data, you need to map that onto what type of endpoint you expect. So it's very different type of analysis, or many other questions will be affected by whether you have a continuous endpoint or a categorical, say binary endpoint. A survival endpoint has very different requirements than those first two, or if you have a count endpoint or a composite endpoint. And basically have a think about how the data you're going to measure maps onto that type of endpoint, and then what kind of estimators those endpoints will measure onto themselves. So if you're doing a survival analysis, you might be using a hazard ratio, but you could also use a restricted mean survival time. For proportions, you could be using odds ratio, proportion difference. And you should be at least having a think about like how you're going to map what you collect onto what type of data it'll create, and then what's your estimator going to be, basically, and kind of comes into the estimate strategy as well. But basically, you should have a consideration early on of what you're kind of going to be using as your criterion for success, effectively, from a statistical, like as a model output, effectively. And then you should also be thinking about like your hypothesis construction, and some of these considerations effectively kind of meld together. I'm just kind of separating them out for convenience. But basically your primary hypothesis of interest should be well defined. So in most clinical trials, confirmatory, certainly at phase 3, your primary interest in is superiority, also known as an inequality hypothesis, but superiority is probably a more accurate term. But obviously in other contexts, you may have a different consideration. So an equivalence trial, the most common scenario there will be bioequivalence, where you want to get a new generic version of a drug approved, and you just need to show that in terms of the PK type measurements, AUC, Cmax, that those give equivalent in terms of the kind of basically the blood concentration. Effectively, you just need to be able to measure that the blood concentration of your drug is the same as the current standard drug, the reference listed drug, because the assumption is if you have the same drug, the generic version, and it has the same route of action, that if it's the same blood concentration, that it should have the same clinical effect, even though the clinical effect is never itself directly measured. But then you have like non-inferiority studies. So for example, if you had a currently intravenously injected drug, but then an oral version was created, that may justify a non-inferiority hypothesis where you don't need to show that the oral version is better, but simply that it's no worse than a specified margin than intravenous route of version of the particular therapeutic. And then superiority might be a case where you need to show it's not just superior in the sense that it's like the null hypothesis that they're the same and that you're trying to reject that, but actually you need to be better than a specified hypothesis. So for example, in a vaccine trial, technically the superiority hypothesis for the vaccine efficacy being 0, that at least someone benefits from the vaccine. But in most vaccine trials, you'd be expected to have a vaccine efficacy of say 30%. That was, for example, in COVID-19 vaccine trials, and that's basically a slightly technically different version than superiority. It's called super superiority or superiority margin, and it's much more related to a non-inferiority hypothesis. But basically, you're basically specifying a margin that's away from kind of the classic null hypothesis of a difference of 0 or a ratio of 1 or a vaccine efficacy of 0, something similar. And then you also have to think about, okay, well, I have an endpoint or endpoints that I'm measuring. Like if you have multiple endpoints or multiple tests that you want to do, you need to think about like, how am I going to decide what a true win for my trial will be? And I'm going to talk here about like multiple independent endpoints, but also there's a whole composite endpoint thing, which is a huge matter of debate right now. We don't have time to go into that, but composite endpoints basically kind of fits into this a little bit as well. But basically you might have a situation where, if you have like 2 things that you're measuring, let's say you have PFS, progression-free survival, and overall survival in a survival trial. Do you need both of those to be significant to find for efficacy that your drug will be approved by the FDA, for example? That's conjunctive power or a conjunctive hypothesis. Do you just need one of them to win, or is it just OS needs to win? Or that'll be a marginal if it's just OS. But if you just need either of them to win, that'd be disjunctive. And of course, you might have like a graphical approach to have the hypotheses string together as well. There's a lot more complexity there once you want to get into it. And as I said, like that comparisons, if you have multiple comparisons, if you have multiple endpoints, you need to have a strategy for that, and then think about, do you want the family-wise error rate or the FDR to be main endpoint, and obviously some negotiation there with the relevant stakeholders from kind of point 1 here above. And then you have trial design considerations, like which trial design can provide the information required. Like, a parallel trial provides you good information about the relative effect, but it doesn't provide much information about, what the effect would be over time, for example, within a subject. Or if you want within subject information, you might need a crossover trial. A single arm trial obviously doesn't have any comparison to placebo, so you don't really have the relative effect. You're putting a lot of trust that the standard that you're going to pair your one arm trial to is actually accurate, and that the inclusion exclusion criteria or just temporal effects or some other effect means that previous data, like based on the database, is still comparable to the current status quo. Then you have multiple arm trials or complex factorial trials, and really you just need to think about like, what do you want to do? Now, in most say phase 3 trials, we're only interested in showing our drug is better than the standard treatment, and so a parallel trial is perfectly adequate for that. But in say a bioequivalence trial, you may be expected to have a crossover component to ensure that you can show that if you give the ORLD and the crossover, that if you switch them AB or BA, that you get the same treatment effect, the same treatment profile, that there's no kind of complication caused not there. And obviously crossover trials provide a lot more within subject information than parallel, but they do have certain constraints. For example, obviously you need to be able to go A, B, but obviously if doing A basically gets the treatment to go away, like say in a vaccine, it's very hard to then give B, because obviously the disease at that point has already gone away or can't be gotten. So crossover trials aren't suitable for certain types of trial, or much more difficult for certain types of trials, say survival trials, and they are for say a continuous endpoint. And then you have to think about like, how does your trial design affect the efficiency and constrain those possible inferences? That's basically going to get into that first point. It's like, think about, well, what type, there's multiple levels to the variance, like which levels are of importance to you, and you get more information from a parallel trial than you would from say a cluster randomized trial, but you get less information than you would from a crossover trial in terms of kind of the variance components, if you want to go to that level. And then if you do need to do randomization, which is obviously nearly going to be true in most parallel trials, so you have the classic RCT, double-blind RCT is kind of the gold standard of evidence, and then you need to think about the randomization method you're going to use. Is it going to be block randomization? Is it going to be just pure randomization, some other method? If you're using block randomization, should you include those as covariates? That's slightly getting further into the considerations, but it's something to keep in mind. And then the randomization level. I think the randomization level is a really important concept. If you're randomizing at the level of subjects, so like in a parallel trial, that's where the inference goes. If you're randomizing the say level of sites, so like you have 10 schools or 10 hospitals, 5 of them get treatment A, 5 of them get treatment B. That's called a cluster randomized trial. And basically, it's a huge point made by Stephen Sen, and it's intrinsic to the analysis done in GenStat, for example, which is statistical software, which is based on this. But basically, randomization as a rule of thumb the statistical analysis should follow the randomization level. So wherever the randomization is, that's where your statistical analysis should be focused in on in terms of your degrees of freedom, in terms of your p-values, etc. And then, of course, we're not going to get too much into logistical aspects today, but if you're doing a double-blinded controlled trial, then obviously, you need to think about the logistics of ensuring that the blind is maintained and think about things like the data monitoring committee and stuff like that. And so then we get into like the estimate or effect size estimate, and you see some of these are kind of playing into earlier points, and I really like, the further down you're going, the kind of further down the chain you are. So things earlier on are really feeding into these directly and indirectly in several ways. But basically, you have this idea of the estimate strategy, which is obviously summarized in ICH E9 or 1, and all of the ICH, the International Council for Harmonization guidelines, which are basically the guidelines created in agreement between the main regulators in the world, the FDA, the EMA, etc., which basically agree, here's how we think clinical trials should be run. E901 covers this thing called the estimate strategy as basically an appendix to E9 itself, which is about the statistical considerations in clinical trials. And it talks about this estimate strategy, which is basically a strategy for thinking through like how we want to create an endpoint or a measurement that will actually answer the question that's of interest in the trial. And basically the estimate feeds into the estimator, which is basically the thing that we, like a kind of our statistical concept we're going to measure as our effect size, and then the estimate is the actual thing we get from our trial. And we might run over several different estimate strategies, treatment, while on treatment, hypothetical. Note that this language is kind of displacing some of the traditional language, like say intention to treat. So be aware of that. And basically it's something you should definitely be aware of. And estimate strategy also places a huge emphasis on what's called ICE or ICEs, which are intercurrent events, and that could be things like dropout, but also could include people crossing over, say most commonly from the control group to the treatment group. Which I believe is called drop-in, colloquially in many cases. So, you need to think about what your estimator should be. So like say the hazard ratio, is that appropriate for my study if non-proportional hazards are there? And then talk about how robust it is to certain assumptions. So it says in a survival trial, the hazard ratio may not be appropriate if you have non-proportional hazards, maybe RMST might make sense there. That's a whole active area of debate, but at least be thinking about these questions. Now the estimate concept is basically a whole webinar by itself. And we did have a opinion webinar from Dr. Luis Rojas a couple of years ago, which is in our webinar archive, that covers it in detail, and he takes a more critical view of it. But yeah, it's a huge area, definitely one to keep in mind. And then only after we got all to that, we even think about the statistics, like what is our statistical model going to be, and that's obviously feeding in and being based on that endpoint choice, the trial design, the estimate strategy. And then we're thinking about like, well, if there's a set of option models or tests that are available for a particular scenario, like how are we going to compare these? How are we going to choose the appropriate one? And that's where, say, power calculations or simulation can be very useful to then establish, is this method going to have more power than this, or is this method likely to be more efficient in terms of cost or time than the other one? And that's all these kind of considerations should be front of mind when considering how to design a clinical trial. And then, of course, you should also be thinking about, well, like this is what I hope will happen or what I think will happen, but what if that doesn't happen? What if there's a discontinuity, like if a trial gets delayed for 6 months? What if the type of data gets more extreme in terms of its variance than expected? And then you're thinking about like how robust is your model choices to this misspecification, to trial deviations, to like a lot of dropout occurring when you didn't expect it. And then if you have options and tuning parameters within a model, you can compare not just across models, but within each model, like this model with this, say with a Bayesian analysis, choosing different priors, for example, would be an example of kind of a tuning parameter or an option, even if you're choosing to do a Bayesian analysis, what is the prior we're going to use? And so then we get into like, okay, this like 7th point advanced trial aspect, you may even want to put this above 6 or in conjunction with 6. But basically, if you're not just going to run a kind of classic double-blind randomized controlled trial, which is fixed term, which is say you start the study, you gather your data, you do the analysis. If you want to start doing things that are more complex than just then running a t-test or something like that, you need to think about like adaptive design. So like what adaptations are you going to do? Like what decision criteria will you use to justify when an adaptation happens? And in a clinical trial, especially a commercial one, you'll probably be expected to pre-specify all this before in the protocol, before even running the trial. And like how does this affect the logistics of the trial? So for example, this has a huge effect on the expertise and the choices available to the data monitoring committee, who may need a statistical group to help them. And for example, there was a really great webinar by Berry Consultants, their interim podcast. It's a really great resource. I would definitely recommend it, which was talking about many of the logistical challenges that come with an adaptive design and what effects it has on the kind of monitoring committee, the people who obviously, if you have a double-blind trial, the people actually running the trial can't do the adaptations for you. So you need an independent third party who looks at the data and not only makes safety decisions like a classic DMC, but is now also making decisions whether to stop the trial early or to add sample size or to drop an arm or to enhance enrich a certain arm and kind of choices like that. And then how are you going to justify if you're going to do a more complex design and the kind of additional logistical challenges with that, like having a more complex DMC and the difficulty that come in maintaining the blind logistically in that case, like how are you going to justify the value for that? Like, are you going to show that the sample size lower, it's going to happen quicker, or some combination thereof? And then if you have Bayesian aspects, like how are you going to define the prior and how are you going to justify using a Bayesian analysis? Are you going to have to verify the type 1 error? So for example, if you're going to use a Bayesian method in a phase 3 trial, this hasn't come out yet, but I think the FDA's Bayesian guidance is expected this summer. So there's a chance that we'll probably still have type 1 error as being a consideration for approving using a Bayesian method, at least outside of certain circumstances like rare diseases. That's my expectation. I did a preview webinar in January that kind of talked about what I expect from that guidance. But that would be my expectation for kind of standard trials, phase 3. And so, like, how are you going to make these decisions and kind of justify this? And like, what value is the Bayesian method going to bring compared to just doing a classic frequentist analysis? And then if you're going to do something that's a little bit outside of the norm, if you're going to push the horizon of what's done in clinical trials, especially commercial clinical trials of phase 3, like how are you going to engage with stakeholders? And all of the guidance we saw says, especially the adaptive guidance from 2019 from the FDA, it's like you need to understand the requirements, but you also need to understand their preferences, and those preferences are stated very clearly there. Simulation is a very good way to verify the operating characteristics. Early engagement is actively encouraged, and there's like things like the CID program, the Company Innovative Design program, which have really pushed that forward. And so, I think pre-specification, simulation, know these concepts, know these ideas, and have those ready to go before even considering doing something more complex, in your clinical trial. And then you get all through all of these things, you might then go, okay, well, how big does a trial need to be? Sample size. Now, sample size was covered in a webinar last year. Everything you need to know about sample size in detail. So I'm not going to cover it in too much detail today. But basically, you need a trial that's big enough to answer questions. So in investigator-led trials, there's still many trials, many studies that are done that don't have enough sample size, that have low statistical power, that have type S, type M errors, using terminology of Andrew Gelman. Type S errors are where the sign of the effect that actually exists is different than the sign your trial has, because the variance, because the power is so low that basically the outcome could basically a random variant could happen anywhere, more or less. And so if you're practical, but like if you're in a trial and you only have so many subjects available, say in rare disease, or you just only have so much budget, at least think about, instead of going like doing some sample size theater and just making things work so it looks like your sample size is justified, at least think about then like what's the minimum effect size that we can actually find. Like if the minimum effect size we can only find is like a hazard ratio of 0.5, and any hazard ratio that's closer to 1 than that wouldn't be findable, wouldn't have power, then maybe you should consider if the trial is viable at all, or whether the trial is even worth running, because bad research can create so much noise and create so much harm in the long term, that we basically need to avoid doing bad trials. And there's a famous quote by Doug Altman talking we need to do less research and better research effectively. His full quote's actually a lot better than that, but basically basically a huge emphasis on quality research, even if there's less of it, is far more valuable than a whole bunch of research where a whole bunch of it is basically statistically spurious due to things like low power. And you should also then think about, if you have a target sample size, what are you going to do to ensure that you can reach that sample size? How are you going to maximize and track your recruitment? And how can you ensure that as few people as possible drop out of the trial? And that's where the kind of patient-centered approach can be very important. So in terms of common pitfalls, and you can see these in trials all the time, the big one, and this was a big emphasis in my sample size webinar as well last year, but basically simplifying your data or simplifying your model. There's a big temptation sometimes to just kind of go, I want something that I quote unquote understand. But oftentimes this is done at the cost of both the interpretability, but also the viability or feasibility of your trial. And so the big one that I often emphasize, and this kind of comes from Stephen Sen, and this is coming from like over a decade ago now, papers that talked about this idea of dichotomania, this kind of mania to take continuous variables and turn them into binary variables, but also similar concepts like taking, multi-categorical variables, so like say Likert type scales data and turning into binary data. And this is taking an idea of like continuous or ordinal data and taking it as a yes or no, or success or not success. Basically, this is a huge waste of research, of time, and a huge waste of your statistical power. And so you can show this idea of the Pitman efficiency, that you're basically losing a third of your efficiency. And this has a dramatic effect on your sample size calculations. I'll show that in a moment. But basically, you could be talking about like 20, 30% increase in your sample size compared to if you just for using a binary test, like say a chi-square test, after you've basically split continuous data half. That's the best case scenario. That's the most efficient split. Say if you knew exactly that the means are like 0, 1, and you split it 0.5 of your two, then that's the most efficient split. And even in that case, you're losing a third of your efficiency. So this is a huge error and something you should definitely avoid. And it has basically its cousin called responder analysis, but not only has the problem of dichotomization, which is basically intrinsic to most responder analyses, but often is misleading in the sense responder analysis often has an arbitrary threshold or thresholds above which someone has become a responder. And if that's based on like a change from baseline type variable, you'll often see these kind of really weird scenarios where you have more power for like specific endpoints. And Stephen Sen goes through this in a lot of detail in the papers that are provided in the references, which goes into dichotomization and responder analysis and why they have so much problems. And you have similar concepts like numbers needed to treat, which have similar issues as well. So basically, the main point you should take away is that if you have a continuous variable or you have a complex ordinal variable, use that data as close to as it is as you can, because there are other examples. So for example, count data is often treated as continuous data or survival data, but if you're using that way, all the counts after the first one basically more or less get thrown away or all kind of get simplified down. So there are other examples, but that kind of dichotomization is kind of the main thing. And the basic principle is use the data you're going to measure, try and analyze as close as it actually is, than trying to force it into a box that you feel will be easier to explain to some other stakeholder. And then there's other kind of common mistakes. So like this has obviously been a focus of the FDA guidance in recent years of like covariates. So if you're going to measure additional information about patients, whether that be simple things like age and gender or some more complex endpoints, something that requires say blood draws or something like that. So if you're going to measure something, you should be then using it, because presumably if you measured something, you presumed it had some informational value, that it added something to your analysis or could add something to your analysis that wouldn't be available if you didn't do that. So for example, if you're doing block randomization based on say age, gender, and smoking status, you should be including those in your model. And not only because it will make your model better in terms of the conditional estimate, particularly if it's a continuous endpoint, but also because it will increase your power. You will have more statistical power for the continuous endpoint, basically because you're taking more variance away from the unexplained, the MSE. But even for other cases like logistic models or survival Cox models, they're still, on average, much better to include your covariates than not. Now, the reasoning for logistic and Cox is a bit more complex. Basically, it kind of gets the estimate closer. I guess the estimate's a bit subdued effectively for the unadjusted model. But the basic principle that covariate adjustment is usually recommendable is there, and you can see it in FDA guidance is provided in the references at the end, that this is something that we should be using a lot more of. And as I said, it increases your power, so why not use this? Now, the one thing that I would say is don't use covariates that were measured post-randomization. So if something is post-randomization, do not include it effectively. So don't get into that type of stuff. You're talking about covariates which are pre-randomization. Yeah, except with the one exception would be probably the baseline, which you can consider to be maybe pseudo pre-randomization. But like most baseline measurements, so the baseline measurement is usually taken before randomization, but that's probably okay post-randomization. But most things, if they're measured post-randomization, do not take those into account, because those are usually going to basically take information that happened during the inferential stage and kind of bring it back into the model, and that can create biases that are very much unwanted. And the one thing I would say is that, like, you may think, oh, I do adjustments via change score, so that's the difference between the baseline measurement and the final measurement. That actually doesn't do anything. That's effectively doing analysis as if you were just using the final measurement. If you're going to use a covariate, or if you're going to use baseline, for example, use it as a term in your model, in an ANCOVA model or regression model. That is how you get the higher power. That's how you get the more accurate inference, in the opinion of most authors. Don't just use a change score. That basically does nothing. Other considerations, so not justifying understanding design. So you should be, if you're choosing between different design considerations, you should be understanding it and comparing them in terms of the robustness, in terms of the efficiency. So you should probably be doing simulation, is probably the recommendation here, especially if you're doing anything that's very much out of the box. But there is often a tendency to kind of just take an out of the box solution that's been used in the previous trial and just use that haphazardly without even thinking about it. You should be evaluating, thinking about, or at least talking to someone like a statistician or to another expert, like why this decision, why this statistical model, why this design is the best design for the questions you want to answer, and also make sure you understand what the outputs of your trial actually mean. Like understand what a hazard ratio value is, understand what a p-value is, understand that your hypothesis made sense. So there's so many trials where they run a superiority hypothesis and basically you get a non-significant p-value, and then they would say something like, well, we kind of shown that these two drugs are equivalent or that they do the same thing. And that's not what you've proven by doing that. That's only provable if you do an equivalence hypothesis directly. And so, you've just failed to reject the null hypothesis. That's not the same as accepting the null hypothesis. And I know there's kind of statisticians going to get into this stuff a lot, but it is very important that you can't just say if you fail to reject a superiority hypothesis that you found that the drug is ineffective. That's not the same thing, or that it's equivalent. Those are different concepts that have their own specific type of testing for them. And I would say that, there's many cases where, with more sophisticated methods, we've got much better allowing users to kind of start with a certain hypothesis and switch to a different hypothesis if they need to. Now, this should be done with care and obviously in conjunction with that actually having some clinical utility to it, but like a kind of a treatment switch from where you start with non-inferiority and then prove superiority, like where non-inferiority is like a lesser burden, get over that hump, now let's prove that it's actually superior. These things are available out there and are of interest if you do want to answer multiple questions or have the possibility of answering multiple questions within the same trial from a statistically valid point of view. One big thing, don't try and change your trial or post hoc rationalize your trial. So pre-specify as much of your trial as possible. Now, if you're doing a commercial trial where you're trying to get a drug approved, pre-specification is one of those things which is non-negotiable with most regulatory agencies like FDA. Like they will accept that sometimes things will happen post hoc that need to be dealt with on a case-by-case basis, usually in conjunction with the data monitoring committee, but they are very, very strongly saying, you should be able to pre-specify anything that is predictably likely to be of interest in that. And so protocols for clinical trial are likely to be extensive, and you should take account of things like the consort and spirit guidelines in terms of thinking about how you're going to design your protocol and then report your trial to the FDA or to the DMC. And then obviously, it's more of an issue in investigator-led trials, but you don't want to do p-hacking, which basically just run 100 different analyses. You see the famous XKCD trial comic, which goes through jelly bean colors and kind of, oh, green jelly beans are significant after testing 20 other jelly bean colors for being significant. Don't try and run multiple analyses without appropriate adjustment. Don't do data dredging where you just go through several different subgroups and find the one that you believe is effective. Like that information can be useful for maybe future trials, but saying that that's proof that one works is usually very, very invalid, because those kind of basically interaction effects in a model, which is what you're looking at there, are very hard to prove and require very specific designs, usually involving crossover if you really want to answer them in a statistically valid way. Don't tinker with your model or change tuning parameters to get the right result for yourself. And I said, don't include post-randomization covariates in your model. And I think it's probably the biggest point that this could have been first or last for me, but if there's other stakeholders, especially ones who understand clinical trial design, get them involved early and often. So that's the FDA. Like the FDA is much more open about dealing with this if you're doing a commercial trial. And that's a phase 1, phase 2, phase 3. They want to have these questions answered because they realize that clinical trial design is evolving and changing very fast. And they want to know what's happening in real trials so then they can inform the guidelines that can help future trialists make the best choices for their trial going forward. But if you have a statistician, involve them at the trial design stage. There is nothing a statistician hates more than basically someone's gone off, collected a bunch of data, bring a statistician in and ask them, can you get me a p-value out of this, or can you get something like that out of this for me? And a statistician basically will hate that, because they'll basically look at this data, see it's a mess, and this is even putting aside things like really color-coded Excel sheets and stuff, but just the data itself, if it's collected in a statistically or in a very poor fashion, like if there wasn't randomization or other considerations, or if you didn't try to ensure blinding and stuff like that, there are likely to be intrinsic biases or low quality or measurement error that makes it almost impossible for you to extract useful inferences from that. Whereas if you talk to a statistician or other data professional early on, they can talk you through the options and create a design that makes it easy as possible and as likely as possible that you will get useful information out of your trial, and information that they can then use, or that you could even use yourself using a standard package in SAS or SPSS, that will be at least useful to some extent. Like, a statistician might have lots of ideas of how to make the best trial ever, but they will most likely give you, here's how a good solid trial would work, like a standard RCT type approach. And here's how you could minimize some of your costs by, say, including covariates in your model in terms of increasing your power. And basically said, like, there's this famous quote here on the right from Ronald Fisher, basically talking about bringing a statistician at the end is like bringing someone into for post-mortem examination to say what they died of. So you're not really helping at that point. So I'd say include them early. And I think that's also true increasingly for including patients. Like, if you can have patients lined up, you have like a group ready to go of patients who are already enthusiastic about going into a trial that you're planning to run, in a particular, say, clinical area, or because there's a certain therapeutic coming up. That will also make it much easier for you to get trial recruitment going faster, and also means that if they're informed and a well-informed, well-considered group up front, then you probably have higher adherence, lower dropout as well. So I think patient, it's not obviously an emphasis here, but patient input, taking into account patients, is really something that will really help you increase the quality of your trial. I see a lot of really great research, especially over the last 5 to 10 years, where groups are talking about strategies and mechanisms to get patients involved and to reduce dropout. But the biggest thing you should do, and it's one of the big recommendations, is to have early and honest engagement with your patients. Like, don't treat them as like they wouldn't understand how a clinical trial works. Talk it through them, talk with them early, try and create a group of them as early as you can who can be recruited to your study, and your trial will likely go a lot faster, a lot better, and you'll end up with more valid and less issues around worrying about multiple imputation and things like that for dropout strategies. And so I think what I want to talk about now is just like power as a clinical trial design choice tool. So power is obviously a useful a priori metric to evaluate the chances of success of your trial. Now, there are similar concepts like assurance or cost decision theoretic approaches, or just cost benefit analysis, conditional power that can be useful in other contexts. But power is obviously something that people are generally used to because it's used as a common criteria for sample size calculations. And so power can be used basically to evaluate across different design choices and different scenarios to illustrate, for example, the cost of doing some of these design pitfalls. And like an obvious way to explain to someone why we should, say, analyze our data as continuous rather than as a responder analysis based on like an artificial dichotomization is, well, the sample size is going to be higher, and I can show that directly in nQuery here today. And standard power calculations with the same inputs with different methods are often an easy way to just verify the effective design choice for other stakeholders. Like if a decision is going to lead to a higher sample size requirement or lower power, that's usually relatively easy to explain to a clinician or to a patient or to some other, maybe say less statistical audience. And so, higher sample size means more cost is generally something people want to avoid. So if you can show that power is one criterion that can display that option, it can be useful for that purposes. For more complex scenarios, so like say adaptive design, power is useful, but you may also need a more holistic view of the trade-offs. So that's where simulation is basically almost required if you're talking about very complex adaptive designs, which they have multiple adaptations and Bayesian components. But even for simple ones like group sequential design, they can still be useful. And you may also need to be worried about other considerations than power, like type 1 error rate, and say how much gain are we going to get in terms of like lower sample size, quicker trials, based on that. And so what I'm going to do here is do like a 2-sample t-test example and then show some variations on the theme that we can get from there. So what we're first going to do is just do this calculation so we know we're on the same page, and we'll move on from there. And so this is nQuery for anyone's aware, and we just had our 9.5 update go live last week. And one of the main updates that we made is this table select overhaul. So the old version is available in the options menu for anyone who wants to keep the old version, but the new version is hopefully going to provide you a much easier mechanism via which to find the specific scenario for design, and also order the options in such a way that you're most likely to get the option that we would probably recommend if we were kind of giving a consulting role for you. We don't do consultations, but if we were to consult, this is kind of what we'd recommend. And so, for example, we have multiple different starting points. And we think about the kind of choices that we had in the first section. Like, we might talk about what kind of design we have. Do we have a fixed term design, keeping it simple, adaptive design? Are we going to include Bayesian considerations, and what effect does that have in terms of our sample size calculations across all three? Or maybe we think about the endpoint. Like, do we have a continuous endpoint, means endpoint, proportions, a categorical endpoint, survival endpoint, even a count type endpoint? Are we going to be using a regression model? So we want to think about like in terms of our model instead of our endpoint or a type of design. And then maybe we want to think about our hypothesis like non-inferiority or equivalence, or maybe we want to base the sample size on the interval width. We've had webinars about that. All these kind of areas have had webinars in our series. Go back, so you can go back to our webinar series if you want to know more about these. And then even like a focus areas like an nQuery, like you might want to think about, correlation agreement type measures, mixed models, and those type of options there. So we'll keep it simple here and we'll think about, okay, we want to just do a classic fixed term design. So we click fixed term designs and we're like, okay, our endpoint in this case was continuous. So we click means, which is basically analogous continuous for our purposes here. Note that means also includes medians, basically non-parametrics as well. In this case, we were doing a phase 3 trial. So obviously the most common choice there is a 2-group trial. And then we have inequality, which is also called superiority. So we usually use inequality because it's a more precise language and less confusion with super superiority, but inequality and superiority are effectively usually used interchangeable. So we click inequality there. And then in this case, we were just using a simple t-test, which obviously is kind of in the same family as a z-test. And the first option here is MTT0. So, we assumed equal sample size in this case. And so we have our significance level of 0.05. And then if we go back to our slide here, we have our means of 12.7 and 15.3 for a mean difference of 2.6. Or minus 2.6, the direction doesn't matter for the t-test. Then we have our standard deviation of 10.6, which gives a standard effect size of around 2.245. And then we have a power of 80%. And we get sample size of 262, which obviously agrees with the original calculation. So this was obviously a fairly simple trial from the New England Journal of Medicine for Sebastian. Very simple design, and they even did a little dropout calculation here. So if we take 524 and divide it by 0.97, this is a classic dropout calculation. It was more emphasis in the sample size webinar if you want to know more about that. We get 540 patients as the actual number of subjects that they recruited there. So in this case, they had a hopefully relatively well run trial where most patients were going to stay on throughout the trial. And so this is a very simple trial, a very simple example. I think most people would be capable of doing this kind of sample size calculation. But now let's think about what other strategies could they have considered to make their trial better or potentially make it worse. And that's where we get on to the next slide here, which is just kind of design, choice, power summary. Now I've used dichotomization here. More accurately, I should probably use simplification because, as I said, you could include taking an ordinal variable and turning it into a binary variable, or taking count data and treating it like survival data. But we're just going to focus on dichotomization as kind of the classic and most commonly mistaken use of simplification of your endpoint. As I said, you should be doing what this trial is doing, which is basically using the continuous data as continuous data. But many trialists might go, well, if your mean response, I think ventilator-free days here, was like less than 13, that's less, not a response, that's bad. And if it's more than 13, that's a response, that's good, or some other thing. Now in this case, because we have a sample size calculation where we kind of know the true control rate and the true treatment rate, or at least what we expected pre-trial, we can kind of pick, okay, the optimal split point between these would be halfway between these two values. So we could find that by just taking 12.7 plus 1.3, so 14. And so basically above 14, good, below 14, bad. But we can easily use like normal distribution theory to basically calculate what that would create the expected proportion in each group. And so what I've just done here is some simple code in OR where I've taken 1 minus p norm, basically the average of these two, so this will equal 14. And then basically I've given that for the case where the true mean is 12.7 with standard deviation of 10.6. And then in the second line, 12.7 plus like basically the same average, but now the true mean is 15.3. So these two are both exactly the same, except I'm using the p norm, which is the cumulative distribution function for the normal distribution, with a mean of 12.7 and 15.3. And basically what you just need to know is that assuming that all those values are true, so assume that the mean in group 1 is 12.7 with a standard deviation of 10.6, and that the mean in the treatment group is 15.6 ventilator-free days with a standard deviation of 10.6, we would expect if the split point is 14, or the average of the two of the control and treatment ventilator-free days, that to be around 0.4511, so around 45% of people in the control group would have a response, and around 54 or 55% would have a response in the treatment group. So we'd have more response in the treatment group than control group, so that's good. But if we just take these proportions, what effect is that going to have on our power or on our sample size calculation? So we'll just do a sample size calculation because that's often easier to explain. And so, I can go back into this endpoint, and maybe in this case, I basically start with the proportions endpoint. So I use endpoint instead of design as my starting point, and I go to say 2 groups, and then I go to an inequality test, and then I might use the simple PTT0. So I just do a very simple chi-squared test. Let's not even worry about other types of statistical tests. So the chi-squared test is pretty classic. And so our group 1 proportion here, we can take from OR, so we've just copy and pasted that across. We go back into OR, we get the group 2 proportion. So basically, as I said, these are the two proportions we would expect if the cutoff was 14. So if the number of ventilator grades is 14, we'd expect 45% roughly in the control group to have a response, greater than 14, and 54 to be group 14 in treatment group. So we do have a response here. And if we put our power of 80%, we can see our sample size per group is now 411. So that's a pretty substantial increase, right? It's not quite double, but it's getting towards that type of magnitude. So if you're explaining to a sponsor or if you're explaining to a collaborator, if you choose to dichotomize, create a responder analysis type situation, you're basically going to have like a nearly double increase in your sample size. Like I think most people would then agree, actually, maybe we should just use the t-test or another type of analysis that uses continuous data like linear regression rather than doing this dichotomization. And I think that's a really useful way to illustrate why this design choice does not make sense and why this is a common pitfall made in trials. That you're basically making you're deliberately making your trial less powerful, less likely to be able to find, conclude for success, to have a significant p-value, even if the treatment that you think exists actually did exist. So we, in this case, are assuming the treatment actually did work by the specified 2.6 increase in ventilator days, and yet now we create a situation where we're much less likely to have significant value, even if that's actually true. So that's obviously a disaster and easy to explain to a collaborator why that might be a bad thing to do. So in terms of covariates, we can basically do a similar concept here. So if I go to covariates and I go to say back into the means case, if I go to the two groups here and go to inequality, if I go to hierarchical design here, you'll see there's an option here called MT39, test for two means in a multi-center randomized design. And basically this design allows you to include the effect of center. It's effectively equivalent to doing an ANCOVA analysis or a linear regression model where you're going to include center in the actual trial. And actually ICC can be shown for sample size purposes. It's not quite as simple for actual real data, but for sample size purposes, you can show that ICC is basically equivalent to the R squared term used in ANCOVA. And of course, you probably know that the R squared is basically the proportion of the variance or the percentage of the variance that can be explained by that covariate or by multiple covariates if you're talking about multiple regression. And so in this case, if we just take the same inputs, so like 0.05, and this is a two-sided level test, just it's given here, mean difference of 2.6. So we don't need to worry about the individual means. We have our standard deviation of 10.6, and then let's say the ICC, basically the proportion, basically the correlation within a center. So the higher this is, the more self-similar people are within each center. So imagine we have hospitals, and we basically are going to randomize within each of the hospitals. So like let's say if it's a one-to-one randomization, each hospital will have 50% people on treatment, 50% on placebo or on control, but treatment itself, the center itself could have some explanatory effects. So, say a public hospital might have slightly different characteristics from a private hospital. If you just have a random sample of hospitals, if you include center in your model, it will on average increase the power of your model, because that would be if you include it as a covariate. If you don't include it as a covariate, you kind of lose that power. You basically have a less powerful model. And so if I just assume, say, a ICC of 0.25, so 25% of the total variance can be explained by the center effect, and then we have a power of 80%. You see our total sample size here is 392, and that's a non-trivial decrease from 524. Now that's a very strong effect. Like I think a more realistic one would probably be something, like maybe 5%. But even then, you can see that we're not seeing a trivial decrease, like 496 versus 524. That's a reduction of nearly 30 subjects. That may make the difference in your trial being viable or not. Now, a note of caution I would give is that in sample size calculations, it's often assumed, at least as a base case, that the ICC is 0, which basically turns this into a t-test. And that's basically done as kind of to avoid the worst case scenario, especially if you're in a regulated context like commercial trial. But if you're doing an investigator-led trial and you need to explain to an author or a collaborator why we should include covariates, this will be useful. But if you just need to, I wouldn't over rely on this, but if you do need to widge the numbers a little bit, having a useful covariate, if you can show, let's say, age or some other biomarker has X% of explanatory power as measured as Y/R squared, you can use that in your model, like let's say I said 5%, and you can get their sample size a little bit lower for the same power. Now, I wouldn't recommend that for a regulated trial if you're dealing with the FDA, but it may be something that might be useful if you're doing an investigator-led trial, if you just need to kind of make the number a little bit. Now, don't over rely on that. That's a kind of one of those kind of tricksy post-hoc things that can very easily be abused, but it's something you can use, and it's obviously very useful even if you're not going to, even if you're going to base sample size on this scenario. It might be useful to talk to authors and say, actually, including your covariate could basically increase your power. So if I change this to a power calculation, for example, and then I use the old sample size of 524, 2% increase in power isn't a huge amount, but it may be enough to kind of get them interested, especially if that goes from, let's say, just below 80% to just above 80%. Once again, that's a bit of a binary consideration, which is what we kind of avoid, but it may be of interest for some types of people who maybe get over that 80% threshold. So sometimes bad logic has to be beaten with slightly better logic. Then randomization level, as you can basically show, it's like, if you basically go to like a paired analysis or crossover analysis, you would expect higher power. But if you go to, say, cluster randomized trial, you're going to expect lower power. And we can easily show that in nQuery as well. So for example, if I go to the assistance menu here and go to standard deviation, and then go from SD1, SD2, and SD correlation. So if we assume that the group one mean, the standard deviations in the group were actually the standard deviations for the pre and post paired consideration, so like pre and post measurements in a paired analysis, and then we say have a correlation coefficient here of, say, 0.5, which is equivalent to 0.25 from a previous example, you can see that the standard deviation of differences is 10.6. So in this case, this is 10.6 for the standard deviation of the differences, but note that's the standard deviation of differences, which is a different concept than the standard deviations of the pre and post, or the per group standard deviation. So if I then go to the paired t-test, so if we go back down to here, paired, and we go to inequality and then t-test, you can see MOT1 here. If I go 0.05, and I'll just use the mean difference of 2.6 again, and the standard deviation is 10.6. So like this is only true for the case where the correlation is 0.5. It would obviously differ for other cases. If we put a power of 80%, our sample is 133. So the sample size is way down versus the parallel trial. But let's use a more conservative example. Let's say the correlation coefficient is only like 0.25, and that gives us a standard deviation of 12.98. But you can see, even with that more conservative assumption, we still have a sample size of 133 here. So this is still way lower than what we have for an equivalent parallel trial. Now, there are things in a paired analysis that just won't make sense for many scenarios. And indeed, if you're trying to measure the relative effect, between independent patients, it might be more useful to do a parallel trial. And obviously, it's impossible to blind, or it's much more difficult to blind this type of trial as well. It doesn't have blinding. So if blinding and that type of thing is important, then you probably going to do a parallel trial anyway. But if a crossover trial, which is a very similar concept, is possible or makes sense, then obviously a paired analysis may be more efficient, and maybe it's a case for an investigator-led trial might be something you want to consider. Especially crossover trials have all these additional information you can get over a paired trial, so I would definitely consider that as well. But that's beyond the scope of today's webinar. But then going in the opposite direction, it's often made that a hierarchical design or cluster randomized design is often used in lieu of a fully randomized trial. So as I said, cluster randomized is where, say, you have 10 hospitals, 5 hospitals, everyone gets the treatment, 5 hospitals, everyone gets the control. That's a cluster randomized trial. But what you'll often see is that those designs have a cost inferentially in terms of and you can show that in terms of reduced power or increased sample size. So if I go to CRT1, which is cluster randomized for 2 means, and you're getting used to these inputs here, 2.6, 10.6. So same inputs as before, but let's say we use the ICC, and we'll use the ICC from this case here of 0.05, because actually the ICC has a very different effect for a CRT trial versus a fully randomized trial that happens to have a center effect. So let's be very clear between here. So this trial is a trial in which within each center, people are randomized. So 50% of people in hospital 1 get the treatment, 50% get control, and that happens in each of the hospitals. In this cluster randomized trial, 50% of the centers, everyone gets the same treatment, 50% of the other get the other treatment. That's a very different design and has substantial effects, and the ICC has a substantial difference then in terms of that. So let's say we had a number of clusters equal to 10, and then we set the power to 80%. Sorry, there's statistical issues why that could be an issue. So what I'll set here is let's say there's 20 people per thing here, and we'll set the power to 80%. So basically, you see here that we have like 27 clusters of 20 here, and that's per group. So you take 27. So we go to the Windows calculator. So if we take 27 per group, so we multiply that by 2 to get the total number of clusters, and we multiply that by 20, you can see that the substantial increase in sample size is very obvious, and that's for a fairly trivial case where the ICC was fairly low. But effectively, what you have here is a situation where a cluster randomized trial would be much less efficient on average than a parallel trial. So if you're going to do a cluster randomized trial, be aware of that trade-off and why you may not want to do cluster randomized trial. If you could randomize it between each center, my recommendation would usually be do, because you will have much better performance, much better power. And then we have group sequential design. So for example, if we go to group sequential design, so in this case, if we're going to say we're going to start with adaptive design, we want to start with adaptive design module here, go to group sequential design, go to means, and go to two groups. We'll use one of the newer group sequential design tables, GST1. And so let's say here we're using the same values as before. I'm actually going to make a small difference here. I won't get into the details, but if you're doing group sequential design, I would very much recommend doing it at the one-sided equivalent level. So if you're doing a 0.05 two-sided analysis, use 0.025 one-sided analysis to just have your initial alpha. So we'll have a tree look design here. We'll have a group one mean equal to the previous values before. We can just copy and paste these across like so. In this case, we specified the two standard deviations, but these are just assumed to be equal. And we'll have a efficacy spending function and a futility spending function. And we'll use O'Brien-Fleming for alpha for our efficacy bound, but Pocock for our futility bound, because usually alpha is fairly conservative, like O'Brien-Fleming, but we may be more liberal with the futility. We want to stop early. We don't want to waste extra resources on a trial where the evidence suggests that the drug is very unlikely to be effective. And then we do a sample size calculation at the usual power of 80%. And you can see we get sample size of 325 per group. So this is a non-trivial increase compared to 262. But let's get further into the detail. Let's consider some of the nuance here. And we can do that by going to the group sequential report here. So if I click expand report, and then I scroll down to these fields here, the expected sample size under H0 and H1, you can see that the expected sample size under the null hypothesis, so where the mean difference is actually 0. We would expect the average sample size due to early stopping. So you can see here, there's like around a 60, nearly 70% chance of stopping early under the futility criterion based on the Pocock futility boundary. So that's around 300. So that means that you're reducing the total sample size from 524 to 300. So that's nearly a 40% decrease in your expected sample size due to stopping early in your trial. Whereas for the efficacy band, because we use a relatively conservative O'Brien-Fleming band, we still got a reduction of around 40 subjects, 40 to 50 subjects, even for a relatively conservative efficacy boundary using O'Brien-Fleming rules. And obviously you could play around with different group sequential designs and stuff like that to get different expected sample sizes, but for nearly any group sequential design, you will see something that looks like this, where the expected sample size, what you think will actually happen in your trial due to you stopping early, will turn out to be much lower, much more efficient than running the equivalent fixed term trial. And so in this case, you're basically getting that huge benefit in terms of relative efficiency. And of course, in nQuery, we provided these simulation tools. So if you wanted to evaluate some kind of interim scenario, so where the mean difference was like half what you expected, you could click through all of these. I'm not going to go through this today. We covered this in several webinars previously, and you could evaluate what happens under those other kind of other scenarios that are of interest to you via simulation as well. And so, there are other considerations, like for example, you could choose between different estimators and stuff, but I think we're a bit low on time, so I'm not going to go through into much detail, but you could say, for example, compare the t-test work to the Wilcoxon test. So, if we were to go back to the table select and go back to the means case, so we go to means, in the two groups, we go to non-parametric instead. There's this table that was added in the 9.5 update, so if you don't see this, you probably aren't the latest version of the software based on the layman method. And we can see here that if we were to rerun our example as is, say using the normal distribution, there will be a slight inefficiency compared to our original example. So we have our mean differences here that we can steal, so 12.7 and 15.3. 10.6 for standard deviation. So the effect size is the same there, power of 80%, sample size ratio of 1. So you can see like 274, 275, versus 262, so an increase of around 10 subjects or so. So the bandwidth U test is not that much less efficient than the t-test, even for the worst case scenario. But the inverse is also true. If you run the t-test in a case where you have non-normal data, say logistic data, you'll see that the t-test doesn't perform that much worse in real terms. So if we redo this example and change the distribution to logistic, but keep the same means, the same standard deviation, if I run this 80%, I get a sample size of 480. And basically I won't get it today, but there is basically papers that show something called the asymptotic relative efficiency. And effectively, you can take this sample size from the t-test and you can multiply that by a certain factor, which I've given this OR code here. Actually, it's like 9.111. So if you divide this by that, you'll get how much worse the t-test effectively does. And so you can show that the t-test actually does basically as well. It's not quite as good. Like this Lehman method does have effectively some efficiency, but yeah, sorry, the man-weighted U-test has some efficiency versus the t-test, but the difference in sample size is within the kind of 10% of the total sample size effectively. So if you take this sample size and divide it, sorry, if you take the sample size and divide it by that 0.9 term, so if we take, say, 0.91, so we take this and we'll take this to the power of -1. Yeah, this is the factor we're multiplying. So if we multiply our sample size here by that, 240, basically the expected sample size for the t-test for the same power of 80% would be at 263. So you can see that both in the normal case, the t-test, like the man-weighted test, isn't that much worse than the t-test, but vice versa, the t-test is pretty robust to non-normal data, and so you don't need to increase the sample size around 20 or so in this particular case. And so, those kind of choices can be illustrated. In this case, the illustration probably says, doesn't make a huge difference to use t-test versus man-weighted u-test. The consideration for using a t-test is probably as much to do with the interpretability of the estimate from a t-test versus the man-weighted u-test, where sometimes it's given in terms of difference in medians, but that's often non-accurate to what the man-weighted u-test actually does. So even though the t-test might be less efficient, if it gives a more accurate or more useful estimate, then you may want to consider t-test. You can show that the effect on power in terms of the increased sample size or the lower power tends to not be too substantial. So it's a useful illustration of that point. So that kind of concludes our point here. I think we've run over time, so I won't have time to go over questions, unfortunately. But please do give those questions before the end of the webinar, and I'll answer all of them via email afterwards. So in terms of conclusions, clinical trial design is this fast evolving area. There's so much change happening. It's evolved from phase 1, phase 2, which have been rapidly evolving over the last 20 years, and phase 3 is increasingly seeing a lot of this change as well. And I think what we want, this has been a response to a real need where we want more efficient trials that have a higher chance of success, which cost less because of the escalating costs of trials over the last 40 to 50 years, which has led to the cost increasing, which means less drugs being investigated than should be. And that means it's more important than ever to understand the principles of good trial design and understand the questions you should be asking yourself by, say, the questions that are in the slide deck, or by looking at the Consort or Spirit guidelines, or by looking at the ICH or the FDA's or the EMA's guidance documents to make sure you're designing the right trial for your study. So you have a study, you have questions, how are we going to design something that answers those questions in the best way, the most efficient way possible? And power can be one way that you communicate why a choice is good or bad. So you can talk about, if I do dichotomization, your power is going to be lower, your sample size requirements are going to be higher, but covariates are going to increase your power and potentially reduce your sample size requirements. These are kind of easy to communicate ways to talk about why you're making a decision that you think should be made for this trial compared to one which maybe is the one that's been used a lot in previous trials, but which you think are inefficient. And then practical examples have hopefully shown that today in today's webinar. Just a brief note, we had our 9.5 update last week, and this added 25 new tables into the base tier of the product, which everyone gets access to. So in the vaccines, bioequivalence, non-parametric, and mixed models areas. And then we added a few new features to the plus and pro tiers. So we added a simulation tool for survival group sequential trials. And then we added 2-stage bioequivalence, a Bayesian bioequivalence to go with the bioequivalence update in the base tier. So obviously plus tier would cover the Bayesian bioequivalence, pro tier would cover the group sequential design, simulation, and bioequivalence 2-stage. And then, as I said, one major thing we're trying to do all the time is improve the user experience of our software. We think this table select overhaul should hopefully make it much easier for you to find the tools and options that are best for your trial. I had a lot of personal input into this as the leader of the statistics team, and I think it should hopefully be a much better experience. But if you have feedback on ways it could be improved or things that you would like to see, that feedback is very much welcome. And you could, of course, email us at info@statsolves.com or go to our website and find the contact details there. And that feedback will be very much welcome. If you have any ideas about what you would like to see in nQuery or in our product, or just want some more detail on anything about sample size, we're always happy to answer those questions. Just note that if you don't currently have nQuery or you have a lower tier of nQuery, you can use statsalls.com/nQuery-demo, and you can try a trial of nQuery with all the tiers, with all the features for 14 days. No credit cards, etc. required, just your email address. And I think you're now required to just specify if you're a commercial or academic type person, but no names or anything like that, just your email address. And we'll send you a link where you can try nQuery demo within your browser without downloading anything. And just to note, the references that I've talked about a lot here are here. This first set of references talks about a couple of webinars we discussed last year or so that cover kind of power and sample size in detail, if you want to get more into the details of sample size calculations and power calculations. And then a lot of useful guides in terms of trial design overviews from basically the ICH, from the FDA, some books from Sen and Harlan and Lakins and people like that. And then there's other ones that kind of cover those specific topics like the trial design downfalls, dichotomization, p-values, recruitment, power sample size division, adaptive design, etc., and the example that was used today from Sebastian. So all I want to say is finally, thank you so much for attending today's webinar. I hope it was useful to you, that there was some useful insights there. Our next webinar will be by my colleague, Dennis Desmond, most likely on bioequivalence, covering indeed many of the new additions that were made in 9.5 update to our bioequivalence suite. So until then, I hope you have a great day, and looking forward to hearing from you in the future months. So once again, thank you so much. Enjoy yourself, and goodbye.